Hey, Assalamu alaikum guys. In this video, we will understand about the phosphorylation and dephosphorylation. The phosphorylation means the addition of phosphate group, while the dephosphorylation means the separation of the phosphate group. So in this way, remember the protein phosphorylation, the DNA phosphorylation, and ADP phosphorylation lead to ATP, a lot more things. So biological significance of protein phosphorylation is important for biological significance is the cell cycle regulation. For example, the cell cycle mitosis and meiosis is possible due to the phosphorylation and dephosphorylation through enzyme. So without this, the life is not possible. Signal transduction is also important for intracellular signal transduction and the enzyme regulation and gene expression. Also, the muscle contraction required the phosphate group. So let's understand about with the model of the cell. So if you will think about that, the primary ligand, the primary messenger can be growth hormone, the insulin, or some other things, which that is the create the signaling pathway. Whenever it will be bound with the receptor of the cell, so in this is the protein kinase activation, and in this the protein kinase is the enzyme group of the enzyme will trigger the protein to phosphorylation by the using of ATP, and the phosphate group will donate to the protein. Like the residues serine, threonine, and tyrosine attached with the inorganic phosphate group lead to phosphorylation. So important thing you should need to understand, the phosphorylase enzyme will lead to phosphorylation while the phosphatase enzyme will lead to dephosphorylation. Remember the ligand again attached I am discussing here. So the protein phosphatase enzyme will lead to dephosphorylation, and in this way the protein again convert into the simple dephosphorylated protein. Anyhow, let's understand about the bigger picture for the signaling pathway to understand how phosphate group will work for the signaling transduction. So in this way, here is the agonist. For example, the different type of agonist are the ligand when bind to become a primary messenger. This primary messenger will lead to trimeric protein G couple protein, which that will separate due to the phosphorylation of the G alpha protein, will lead to trigger the adenylyl cyclase enzyme to use the ATP to convert the cyclic AMP, and this cyclic AMP dependent protein kinase will activate, and this activation due to the dephosphorylation, and this is an example of the dephosphorylation. And in this way the important thing is that this activation will lead to activated catalytic subunit will separate, and this separating will lead to phosphorylation of substrate like DNA will lead to activate and the gene activation. The gene activation or inhibition so on the basis of the requirement. For example, the phosphorylation of substrate, for example the DNA or protein. But first we will understand protein and then we will understand about the other. So phosphorylase kinase enzyme used to activate the phosphorylation of the other protein. The activated phosphorylase kinase will lead to activate the certain type of protein for activation of the DNA, and in this way the DNA will activate to synthesis certain type of the protein via mRNA production, that is the transcription to translation. But this activated phosphorylase kinase enzyme will lead to bind with the phosphorylase B enzyme, lead to activated the phosphorylase A enzyme, lead to glycogen phosphorylation, and the glycogen will separate into the form of glucose 1 phosphate, and that will lead to glucose metabolism, and that is the catabolic cycle. And the glucose will use for the Krebs cycle and glycolysis to produce energy. So this is the catabolism. So in this I remember about that this is the phosphorylation. So let's understand about the dephosphorylation. So this, for example, the ATP contain adenine, ribose, sugar, and the triphosphate, mean the three phosphate group attach very unstable bond. This ATP will provide the energy to, in the form of heat or in the form of mechanical or in the form of kinetic energy, some other things. So in this way the energy produced with the separation of the phosphate group by the using of the phosphatase enzyme. And in this way, the ATP ADP cycle will occur. In this way, the phosphorylation. So in this way, the dephosphorylation means the diphosphate, the ADP will basically ADP will form, and this again phosphorylase enzyme used to phosphate group add to energy absorbed from food. So this is the again conversion of the ATP from the ADP. This cycle will continue with the using of the food in the form of glycolysis, Krebs cycle, as well as the electron transport chain to synthesize the phosphate group by the phosphorylation. Anyhow, the like ATP synthase enzyme. So anyhow, here is the pyrophosphatase enzyme used to double phosphate group separation to produce a lot more energy in the form of heat for the muscle contraction, enzyme working, and DNA synthesis. So in this way, important thing you should need to understand that will become a monophosphate to become a adenylyl cyclase enzyme used in the inside of the cell as a signaling pathway will lead to convert the cyclic AMP. The cyclic AMP should, you can watch that this is the secondary messenger and contain a cyclic phosphate basically attached with the oxygen. Anyhow, this cyclic AMP will trigger the protein kinase activation, lead to cellular processes like the response of the cell because it is the secondary messenger after the ligand. So this is all things you should need to understand. So in this way, this is the dephosphorylation, while the phosphorylation to again dephosphorylation to phosphorylation. So in this way, phosphatase enzyme used to dephosphorylation, while the phosphorylase enzyme used for the phosphorylation. In this way, here is the ATP, which that bond with the magnesium as a basically is the catalytic domain. In this way, for example, the protein phosphorylation will occur with the serine residue, which that combine, and in this way, remember this is the negative ion containing anything which that get the proton, and in this way separation of the phosphate group, while the conversion into the ADP, while the donation of the phosphate group to the phosphoserine formation. Due to the amino acid, serine will combine with the phosphate group. Anyhow, this is the important thing you should need to understand. While on the other hand, the basically is the hydroxyl group release combined with another to become a water or something. So this is the phosphorylation of the protein, while the dephosphorylation of the ATP. So this interconnection to each other, dephosphorylation and the phosphorylation. I hope you make sense about the biological aspect of the phosphorylation and dephosphorylation, which that is very, very significant. Why? Because it is basically important for the cellular processes like the signaling pathway and the cell cycle and the protein activation for the synthesis of the protein and protein activation, and on the other hand the DNA activation for the synthesis of the mRNA and the protein synthesis. So the phosphorylation is very, very important for storing of the energy and the enzyme working and a lot more things. So the phosphate group also act as a cofactor for triggering of the enzyme. Summarization is that the signal in will lead to inactive enzyme to activate, and in this way the activation required the ATP and the protein kinase, and in this way the activated enzyme will work for signal out, while on the other hand the again dephosphorylation of that protein through phosphatase enzyme to become inactive. So this activation and inactivation of the protein, enzyme, DNA, and a lot more things like the ADP will work. I hope you make sense about that. Bye.